In this video we will discuss about the final experiment that is done in this paper, where they explain the mechanisms which dominate the NK-92 mediated cellular cytotoxicity. So here some of the things that we need to understand is that here the cytotoxicity means the NK-92 mediated cytotoxicity which is assisted by antibody dependent cellular cytotoxicity, that is ADCC, which is commonly known as. So here what they did is they took two kind of cells, one that is HER2 positive cells and another one is HER2 negative cells, and in order to trigger this ADCC they use the antibodies that can recognize these HER2 positive target cells. So and for that the target cells that they use that has this HER2 positive properties is BT474, and the HER2 negative cells are this MBA-MD468. So these two cells that they have used, so that they can clearly see whether this NK-92 mediated cellular toxicity affected by which mechanisms. So when they did the experiment with different NK-92 variant that they have developed, that is one variant that has this CD16 cDNA and one variant that has the CD16 CAR NK-92 that has the intracellular domain also, that is 41BB. So when they use one control also which is NK-92 CD713B, and when they did their studies with these HER2 positive cells and in the presence of 10 microgram per ml of this Transduction antibodies that has the affinity towards this HER2 positive target cells, that is Transduction, something like this. You can get the name from the descriptions. So when they use that antibodies, and what they are able to see is that and they check the expression of CD107. So what is CD107? It is usually expressed on the activated NK-92 cells which are ready for degranulations. Degranulations means they will release the granules such as this Granzyme perforin, and so it is well known in the literature the relationship between this Granzyme and perforin, that they will be secreted more if there is more CD107 expressions on the cells. This is what their idea is, to check the CD107 expressions on the NK-92 cells once they are co-cultured with the HER2 positive cells and HER2 negative cells. So in the case of HER2 positive cells, they are able to see an increase in the signals of CD107, and the increase is maximum for the case where they use CD16 CAR NK-92 with intracellular domain 41BB, and that can be seen in the first row of the panel A. And in the second row of the panel A, they have used the HER2 negative cells, that is MBA-MD468. Here although they see is some increase in the activations, but that increase remains almost similar in all the three cases, or we cannot say that similar, they increased, but the increased amount is very low. So once we check the quantification results, then we will appreciate the difference between HER2 positive cells and NK-92 activations and HER2 negative cells and NK-92 response. So first we will let us go through this BT474 results. Here the results, the important thing here we need to notice is about the y-axis. Y-axis shows that y-axis shows the CD107 expressions, and it increased up to 70% for the CD16 CAR NK-92 with intracellular domain 41BB, and this is statistically significant compared to the case where there is no ADCC is facilitated, where there is no ADCC facilitated NK-92 occurs. And however, when they did their studies with the HER2 negative cells, here the y-axis values are only till 15%, indicating that the enhanced cytotoxicity, CD107 expressions reaches only up to a 15% or slightly lower than 15%, and that too for the case the CD16 CAR NK-92 with intracellular domain 41BB. So this value indicates that the HER2 negative cells are less prone to this ADCC assisted NK-92 cytotoxicity because they are not specific targets towards this genetically modified NK-92 cells. And so this is how they validate or how they come up to, and in the supplemental figures they also present the results for other pathways, that is FasL ligand, that we have discussed when we are discussing about the previous papers that also discusses about NK-92 mediated cytotoxicity. So just like the previous paper, this paper also comes to a final conclusion that it's the Granzyme and perforin that dominates the NK-92 mediated cytotoxicity compared to the FasL dead cell receptors ligand. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.